This is chapter 14. We are going to look at DNA structure and how DNA is replicated. DNA replication is just copying DNA. It's making two daughter strands from one parent strand. But let's get started. Oh, before we really get started, this chapter shows a lot of experiments. And it's okay if you don't understand how the experiment was done. The important part in most cases is to learn what these scientists showed, so what the experiment showed. Anyway, so let's get started. Fred Meichner was the first guy that isolated nuclei from white blood cells. He named the mixture that he got out of nuclein. And he learned that it was really high in phosphate. And when we look at the structure of a nucleotide later, that'll make a little more sense. So he kind of hypothesized that maybe the nucleus was a place to store phosphorus, which was false. I don't care too much that you know the outcome of this experiment. They pretty much showed that genes can be transferred horizontally and not just vertically. Vertical gene transfer is like what happens in inheritance. I don't care too much that you know what horizontal gene transfer is, so this isn't too important. This experiment was very important by Hershey and Chase. They showed that hereditary information is on DNA and not protein. So DNA was the macromolecule that carried information for heredity, not protein. That's what this experiment showed. About the same time, Chargraph started analyzing DNA content. They knew that DNA had a sugar, a phosphate, and a nitrogenous base. So here in yellow, it's a little hard to see, is the pentose sugar. If this part of the molecule has an H, it's deoxyribose. If this part of the molecule has an OH, it's ribose. In DNA, this pentose sugar is deoxyribose. In RNA, the pentose sugar is ribose. Each nucleotide has a pentose sugar. Each nucleotide has a phosphate group. And each nucleotide has a nitrogenous base. The nitrogenous bases in DNA were thymine, cytosine, adenine, and guanine. Chargraph learned that when he analyzed the content of DNA from different species, that the amount of cytosine roughly equal the amount of guanine, and the amount of adenine equal the amount of thymine. So basically, he learned this concept of complementary base pairing, which states that in DNA, adenine is always going to hydrogen bond with thymine. They will complementary base pair. and in DNA, cytosine, well, in DNA and in RNA, cytosine will always complementary base pair and hydrogen bond to guanine. And this is known as Chargaff's rule. Another thing to keep in mind, using Chargaff's rule, you can figure out how much cytosine is in a DNA sample, how much thymine is in a DNA sample, how much adenine, how much guanine, as long as I give you the percentage of one. So let's say in a certain species, there is 10%, let's just say cytosine. So in a certain species, if you analyze the content of the genome, of the DNA, and you find that there's 10% cytosine, you can guesstimate what the rest is going to be. Since cytosine has to equal guanine, that's the easy one. Guanine will be 10% also. And the cool thing about percents is they add up to 100. So if cytosine and guanine only add up to 20%, 10 plus 10, we know the adenine plus thymine has to make up the other 80%. That looks like an 86. There we go, 80%. But since adenine equals thymine, that means each has to equal 40%. So your adenine equals 40. And your thymine equals 40. So in this organism, 10% of its DNA contains cytosine, 10% contains guanine, 40% contains adenine, and 40% contains thymine. If you add all this up, 40 plus 40 is 80, plus 10 is 90, plus 10 is 100. We equal 100. So if I give you a number for this, you should be able to use Chargaff's rule to calculate the rest. The last important point about nucleotides is that in the cell they are usually all assembled together. We talk about them as being made up of 3 separate parts. Oops. But in reality, most of the time in the cell, they are, well, free nucleotides are in this form, the nucleotide triphosphate. But in DNA, once it's linked together, it's in this form, nucleotide monophosphate. But that means your phosphate groups, your pentose sugar, and your bases are all connected. They're all hooked together. They're rarely separated in the cell. All right, so that's all for that page. I want to focus on this first before Watson and Crick. Rosalind Franklin was a female scientist in the 60s. She was doing X-ray crystallography to try to figure out the structure of DNA, and that's a good method to figure out the structure of DNA. You don't really need to know how it works. But basically from her data, she learned that DNA is a double helix, and that this helix has certain dimensions. And Watson and Crick, right here, took Rosalind Franklin's data along with Chargaff's rule, and figured out the actual current model of DNA structure. And I'll show you that on the next figure. But basically, Watson and Crick sort of assembled other people's work to get this data, and they're the ones that won the Nobel Prize, even though they would have never gotten their Nobel Prize without Rosalind Franklin. They were gonna award it to Miss Franklin sort of retroactively, but she died before they could get it done, and you can't award the Nobel Peace Prize to someone who is deceased. So that's kind of a shame, but I rather you know Rosalind Franklin than Watson and Crick. She's the one that discovered that DNA was a double helix and the dimensions of it. So here's 3 different representations of the current model of DNA structure. We got a cartoon, we got a ball and stick model, and then sort of a biochemical representation. So yeah, DNA is a double helix. It has a major groove and a minor groove. The outside of the molecule is made up of alternating sugar and phosphate components. So sugar, phosphate, sugar, phosphate. That is the backbone, the outside of the molecule. On the inside in here, that's not showing up super well. On the inside in here, you have the nitrogenous bases hydrogen bonding to each other. And as a rule, thymine always base pairs with adenine, and guanine always base pairs with cytosine. And that's due to their structure and hydrogen bonding. But that concept that adenine always pairs with thymine and guanine always pairs with cytosine is how DNA replication works. It's how transcription and translation works. Transcription's making RNA, and translation is making protein. So all these features of life based on one very simple concept, that thymine always complementary base pairs with adenine in DNA, and that guanine always base pairs complementarily with cytosine. I don't really care if you know how this works. Basically know that if you hear Sanger technique or Sanger method, this was how the Human Genome Project was done, how they sequenced DNA from different species. And now, in 2017, they've sequenced quite a few different species. It's something that can be done very quickly now because it's been automated. Along with the Sanger method of DNA sequencing, you had to do gel electrophoresis. I think I'm going to save this for the next chapter. I'll talk about gel electrophoresis then. Okay. So an important difference between eukaryotes and prokaryotes is that DNA replication, DNA synthesis in eukaryotes is compartmentalized. It's contained in the nucleus. Away from where protein synthesis occurs. Protein synthesis in the eukaryotic cell happens either out on the rough ER where the ribosomes are, or in the cytoplasm where ribosomes are. So in the eukaryote, DNA and RNA synthesis have been separated from protein synthesis. However, in the prokaryote, there's no compartment. So DNA, RNA, and protein synthesis all happens in the same place, in the cytoplasm. In eukaryotes, DNA is tightly packed, especially during mitosis and meiosis when the chromosomes are going to be moved around. This makes sense because we have 46 chromosomes that we have to contain in a nucleus inside a cell. The prokaryote just has one single chromosome, and so it has a lot less to pack. Now let's look at the different levels of DNA packaging. So you can have, it's pretty rare, but you can have naked DNA, and by naked I mean it's not attached to proteins or anything like that. It's just nucleic acid 100%. DNA needs to be in this form in order for DNA replication to occur. It also needs to be pretty close to this form for transcription to occur. Transcription is making RNA from DNA. Most DNA, however, is wrapped around a complex of proteins called histones. And they wrap around like 2.5 times around the histone, the DNA will. And this, just this wrapping around the histones will condense DNA a little bit. The unit of a histone with the DNA attached, wrapped around it, that unit is known as a nucleosome. Nucleosome. And they kind of look like beads on a string. The nucleosomes can be coiled in a chromatin fiber, and that coiling will further pack this DNA. Now, a lot of DNA in a cell's nucleus is in this form where DNA, excuse me, hold on. DNA is wrapped around nucleosomes, and then that fiber with the nucleosomes is coiled. When DNA is in this form, it's very hard for, well, it's impossible for DNA replication to occur. And transcription and translation are probably not going to occur. Well, transcription is not going to occur when DNA is in this form. So, say, let's just take a liver cell. So, the parts of the DNA that the cell needs to be a liver cell, to make proteins related to liver function, those parts will be in this form, or possibly wrapped around a histone, but easily accessible. And then sections of the genome that are related to being a nerve cell, a liver cell wouldn't need that. So those parts of the genome inside a liver cell would be pretty tightly coiled and unaccessible for transcription. Now, before, well, really during mitosis and meiosis, when we say DNA is condensed, this is what happens. So these will be coiled up into these fibers. And then these fibers will be looped and attached to this scaffold protein in here. And that will make this type of fiber once it's all attached. And that's what we see in prophase, or prophase 1 or prophase 2. That is the condensed chromosome. So those are all the levels of packaging that can happen for DNA. Know that it has to be sort of loose for replication, transcription to occur. And it's packaged, or it's more tightly packaged when replication, transcription, translation is not occurring. Okay, let's move on. Know that back in the day there were 3 models of DNA replication. Here, gray is parental or old DNA, and light blue is new DNA that's been synthesized, that's been made, assembled, built. So the conservative model, conserve means to keep, means that from one parental strand, one parental molecule of DNA, sorry, not strand, you would make 2 new DNA molecules. One would be completely old and one would be completely new. So for this to happen in the conservative model, 2 strands of DNA would have to separate. And then you would build new DNA using those old strands. But then when they reassembled, they would reassemble this way so that the old strands are together and the new strands are together. That's the conservative model. The semi-conservative model, semi means half, conserve means keep, is this one right here where the parental strand would split. And then using the parent strands of DNA as templates, you'd make the new strand, and they would just stay that way. So that's the semi-conservative model. And then the dispersive model, one more thing about the semi-conservative model. So in the semi-conservative model, from one parental strand, or parental molecule, you make two daughter molecules, where one strand is old or parental, and one half is new. The last model, dispersive, is kind of the idea that from the parent molecule, this was broken up into little fragments, which were then used to make new fragments. And then they were sort of assembled like this. So they were broken apart and then put back together. That was the dispersive model. This experiment by Stahl showed that the semi-conservative model was correct. That's all you need to know from this. This experiment by Messelson and Stahl showed that the semi-conservative model was correct. So that's what we're going to learn about. Okay. So let's decode this image a little bit. Here they're not really showing you much of a difference between the template strand, or the parental strand, and the newly synthesized one. Just know that the template parental strand is on the outside, and that the newly synthesized one is on the inside. This image is showing a replication fork, which is one half of a replication bubble. So this green circle here is topoisomerase. Well, let's talk about topoisomerase in a second. This blue triangle is helicase. Helicase will separate the double-stranded parental DNA molecule into two single strands. So some people say it unwinds the helix. But basically it breaks the hydrogen bonding between the base pairs. Once it does that, these purple little things, which are called single-strand binding protein, they bind to the single-stranded DNA and keep it from going back to being double-stranded. It keeps it open. Then we can talk about topoisomerase, the green ring here. So as helicase is pulling apart these two strands of parental DNA, helicase doing that would normally create tension upstream from where the replication fork is, and topoisomerase relieves that stress. So far we've gone over helicase, single-stranded binding protein, and topoisomerase. Now when you open up this fork, and for the moment we're going to look at the parental template strands, which are going to be the outer ones. If you look at this template strand, oh yeah, that's one error here. Okay, so if you look at the template strand, on the outside, this side starts off 3', which means if you follow this along, the other side's going to be 5'. And remember, these prime numbers are just directions, so it tells you which direction the DNA is oriented. And DNA is anti-parallel, which means that if you have 5' on this side, you have 3' on the other, they're oriented in opposite directions. And if you followed this back along, this should say five prime. And this should say 3'. Now, this figure isn't in the print book. You have to open up the PDF to look at it. So maybe it's fixed in the PDF, but at least on my PowerPoint slides, the figure's incorrect, and the figure's not even in the printed textbook. So hopefully they fix that, but if not, you might want to make this correction in your book, that this end should have said 5' and this end should have said 3'. Anyway, so let's look at the parental strands. This one out here has 3' on the left, 5' on the right. And this one out here has 5' on the left and 3' on the right. And DNA can only be made 5' to 3'. So for this bottom strand, this bottom parent, since it's oriented 3 on the left, 5 on the right, your DNA is going to be built in this direction from 5' to what it will eventually be 3' over here. The strand that is produced towards the fork, towards where helicase is, that strand is called the leading strand. It's the leading strand because it only needs one primer to begin, and it's made continuously. So even though they don't show it, on this cartoon they show primers with an orange little arrow. So this strand began with a primer. A primer is 5 to 20, maybe 25 RNA nucleotides that are needed to begin DNA replication. And like I said, the primers on this cartoon are shown by orange arrows. I've kind of drawn one down here for the leading strand. And the primer is made by the enzyme primase. And the primer allows DNA polymerase 3 to attach to the parental DNA strand via the sliding clamp. DNA polymerase 3 will just start adding nucleotides one at a time to the 3' end of the primer, and so it will make new DNA 5' to 3'. And how does it pick which nucleotide to add? The way it picks which nucleotide to add is simply by a complementary base pairing. So if there's a T on the parent strand, DNA polymerase 3 will add an A on the new strand. If there's a G on the parent strand, on the template strand, DNA polymerase 3 would add a C, and so on and so on. And it's just going to read the template strand and add the complementary base on the new strand that's growing. Now, this enzyme, DNA polymerase 3, is the main enzyme for creating DNA, for synthesizing DNA. So remember that that is number 3, DNA polymerase 3. That is your major DNA synthesizing enzyme. Okay, so on the other side of this fork, where we're using this template strand, which is 5' on the left and 3' on the right, that means the DNA is going to have to be synthesized in this direction. In this figure, it would be right to left instead of left to right. And you'll notice the replication fork with helicase and single-strand binding proteins. The fork itself is pretty packed. And for that reason, Okazaki fragments are made, which are, I don't know, hold on. These Okazaki fragments are about 200 nucleotides long. Since the lagging strand is made in this direction using fragments, it said that it's synthesized away from the fork, but still 5' to 3', in pieces or in fragments. So once some space opens up, primase will lay down an RNA primer on the lagging strand. And then that allows DNA polymerase 3 to bind and start to synthesize DNA here, which is shown as the red arrow. Remember, the orange arrow here is RNA. And DNA polymerase 3 will synthesize until it hits the next Okazaki fragment, and then DNA polymerase 3 will fall off. Eventually, the RNA primers will need to be taken out of the newly synthesized DNA strand, because we want to all be DNA. We don't want parts of RNA to be in there. And that's why this cartoon probably does not show a primer in the leading strand, because it was taken out a long time ago. But being shown over here on the lagging strand is DNA polymerase 1. DNA polymerase 1 will take out the RNA primer. It'll take out the RNA nucleotides and replace them with DNA nucleotides. And once again, polymerase 1 will read the template strand. If the template says G, it'll take out the C um, RNA nucleotide and add a cDNA nucleotide. If the parental strand says A, it will take out the uracil RNA nucleotide and add the thymine DNA nucleotide. So that's what DNA polymerase does. It takes out the primer and replaces it with DNA. And then in several spots along the lagging strand and even a couple places on the leading strand, you'll have some gaps in the sugar phosphate backbone. And those gaps are sealed by DNA ligase, which is this little blue oval here. And that will eventually make between primase, DNA polymerase 3, DNA polymerase 1, and DNA ligase. Eventually the lagging strand will appear continuous like the leading strand. And these forks move at 1,000 base pairs per second, or 1,000 nucleotides per second. So this whole process happens very quickly. Okay, so let's, so next we're going to talk about telomere. So at the very ends of linear chromosomes, it's very hard to finish DNA replication. The leading strand can go all the way to the end of the chromosome, but the lagging strand cannot, due to the way that it's synthesized. So this leaves a big gap here on the lagging strand. And that would eventually mean that every time DNA is replicated, the chromosome would get shorter and shorter and shorter and shorter, and eventually you would start losing information that's important for making proteins. You would eventually start losing genes, and that could lead to organism failure. So that could lead to death. So we don't want that to happen. So at the end of chromosomes are these repeating segments called telomeres. They don't code for any gene, and these telomeres are repeated 100 to 1,000 times on each chromosome. Now, if you take a cell out of a live body, I would harvest fibroblasts from embryonic mouse hearts back in the day. So say I would culture these fibroblasts in a petri dish. They would divide about 10 times, and then they would just stop dividing. They wouldn't divide any further. And this is due to the telomeres. So basically, after dividing 10 times, these fibroblasts lost all their telomeres, and if they divided any further, they could start losing genetic information that was important, and so they quit dividing. Some cells in your body do the same thing. They only have a limited amount of times they can divide before they stop dividing because they've lost all their telomeres. Now, other cells in your body divide all the freaking time. And their telomeres are maintained by this enzyme called telomerase. And basically, telomerase has an RNA component that can provide a template to repair telomeres. So that's what the rest of this thing is showing. It's just showing that telomerase can act as a template to repair telomeres. And the sequence for telomere in humans is TTAGGG. So here's an intact telomere, TTAGGG, and telomerase is building a new telomere, which will eventually lengthen out like this, TTAGGG, and then it can build another one, and another one, and another one. In this way, the telomeres are never being deleted due to the fact that the lagging strand can't go to the very, very end of the chromosome. Now, in prokaryotes, their chromosomes are circular, so this never happens in prokaryotes. This is just a eukaryotic mechanism. It was thought that telomeres were responsible for aging, and so while we do still think it's related, they did an experiment where they overexpressed telomerase in some cells, and those cells ended up not ever aging, but they instead became immortal and basically turned into cancer cells. So telomerase is not the solution for aging, but it is pretty cool for maintaining the ends of chromosomes. Elizabeth Blackburn was the one who discovered telomerase and worked on its mechanism. Okay, so the last little section of this chapter, we need to talk about DNA repair and mutations. The first type of sort of DNA repair is called proofreading. And proofreading can be done by DNA polymerase 3 or DNA polymerase 1, but the main one's DNA polymerase 3. And basically it's looking for mismatches where the hydrogen bonding doesn't occur. So here's a mismatch. You have G opposite an A, where there should be a T opposite of A. So DNA polymerase will just take out the G, add a T, and move on. And that process is known as proofreading. And it just corrects errors that are happening while replication is happening. Now, even though proofreading occurs, sometimes mismatches will be in the finished product. Now, there are some modifications that happen to adenine molecules after replication has occurred. So these modified adenines can tell your enzymes which strand is the old strand, the template strand, and which side is the new strand or the daughter strand. And that tells it which side is correct. So if you have a mismatch, if these adenines weren't modified, you wouldn't be able to tell which base is wrong, whether the G was wrong or the T was wrong. But since these adenines are modified, the enzymes know which strand is correct. You would cut out the T here and add back a C, which would be correct. And that's shown right here. So they've taken the thymine nucleotide out and added the cytosine nucleotide, which is correct. So that's known as mismatch repair. The last type of DNA repair that we will talk about occurs well after DNA replication. A common result of ultraviolet exposure is this thing right here. It's called a thymine dimer. If two thymines are next to each other, if you add UV light, they can form a covalent bond between the two of them, which means they're no longer hydrogen bound to the adenines on the other side. And this definitely interferes with DNA replication further down the timeline. So the way these thymine dimers are repaired is by nucleotide excision. So these red arrows here showing where the enzymes would cut the sugar phosphate backbone. It would take out this whole section of DNA from cut to cut. And then they would come in and use the opposite side as a template to build new, like a new little DNA patch from here to here. And that way you can get hydrogen bonding between the thymines and their adenines, and you remove the thymine dimer. Now, if you don't repair thymine dimers, you can get mutations in your DNA and possibly skin lesions or skin cancer. Okay, so the last little section is on types of mutations. The first three are going to be point mutations. A point mutation is where you exchange one nucleotide for another. So instead of a T, you put a C. So let me see. Okay. So in this first example, they have changed this A to a T, and that is a point mutation. You do a single nucleotide substitution. Point mutations happen all the time. Several of them are silent. And silent point mutations have no effect on the protein that a gene builds. So proteins are polymers of amino acids, and their structure and function depends on the order of these amino acids. And so in a silent mutation, this little section of DNA codes for serine, valine, proline, tyrosine. And if you substitute this A for a T, there's no effect on the amino acid sequence. It's still serine, valine, proline, tyrosine. Still the same exact sequence. Now there's a missense substitution, which is a point mutation. We're going to change this C to an A, which is a point mutation, a single nucleotide substitution. But instead of having no effect on the amino acid sequence, we're going to change that amino acid sequence. So instead of serine, valine, proline, tyrosine, it's now serine, valine, threonine, tyrosine. And when you change the sequence of amino acids, you can change the structure, you can change the function, you can make a broken enzyme that's always on, or a broken enzyme that never turns on. You can make several different effects on the protein. A nonsense mutation is a point mutation. They have changed this C to a G. And what a nonsense mutation does is, due to the point mutation, instead of coding for an amino acid like tyrosine, you encode for a stop codon. And we'll learn about codons in a couple chapters, so don't worry if you don't completely 100% understand this. But when you insert a stop codon into an amino acid sequence, you can get a truncated protein. And a protein that's half the size of what it should be is probably not going to be functional. I don't care about frame shift mutations just yet. I'd rather talk about that when we start talking about proteins. So that's the last part of this chapter. Know how DNA replication occurs, be able to compare and contrast the leading strand with the lagging strand. Know concepts like anti-parallel, semi-conservative, Chargraph's rules, complementary base pairing. Know what all the enzymes do during DNA replication. Know what a nucleosome is. Know about Rosalind Franklin's work, Watson and Crick, Chargraph, Hershey and Chase. You don't need to understand the experiment, but know what they found. What else? And then know about telomeres and telomerase and the 3 types of DNA repair that we talked about, and know what a point mutation is. Know the difference between silent, missense, and nonsense mutations. The main part of this chapter, though, is knowing all the enzymes for DNA replication and how DNA replication occurs. It's important to know, I don't know if I emphasized this enough earlier, but DNA replication only occurs in the nucleus. It does not occur anywhere else because the nucleus is where the DNA is. Alright, so that's my last point. Good luck.